Assalamualaikum warahmatullahi wabarakatuh and hi everyone. We are from Group 9. Today we would like to present the topic Isolation of antioxidative compounds from Micromelum minutum, guided by preparative thin layer chromatography to 2-definite 1-picrohydrazine PTLC-DPPH bioautography method. Before start with the presentation, I would like to introduce the group members. My name is Ainsya binti Muhammad Abdul Fatah and my metric number is 201888.
Hi and Assalamualaikum everyone. My name is AFRINA MARDHIAH binti Abdul MANAN and my metric number is 204158.
Hi, I'm HAGALYA MATHIYARASU, metric number 203062.
Alright, so I will proceed with the taxonomy for the Micromelum minutum. What is taxonomy? Taxonomy is the science of naming and classifying organisms, also a standard way to refer to a species and organize the diversity of living things. Okay, for the Micromelum minutum, the kingdom is Plantae, the phylum is Magnolia, the class is Magnolepsida, the order is Sependellus, family Ruticae, genus Micromelum, species Minutum, G-force with an arm. The vernacular name is divided into 4, which is Cemumar, Cemama, Limeberry, and Red Limeberry. Cemuma and Cemama is come from Malay, and the limeberry, also red limeberry, is come from English. Okay, then the traditional usage of the plants. They have 4 usage that contain in the plants, which is the leaves, stem, fruits, and also flower. The leaf is used as a febrifuge. What is febrifuge? Febrifuge used to reduce our fever. Next, stem is used as a herb or preparation intended to either prevent formation of gas in the gastrointestinal tract, also known as a stomachache. Then, fruit is helped to constipated patients get rid of his body backup waste. For the flower, it acts as a expectorant. This is used as a cold medicine that helps you to cough out mucus from your lungs. Then I will pass my presentation to my friends.
So now, I'm going to present about my part, which is known as secondary metabolite present in the plant. Firstly, the secondary metabolite present in the plant known as sesame, and the class of the metabolite is lignin. Secondly, the secondary metabolite present in the plant is Micromelum, and the class of the metabolite is flavonoid. Additional note, it is a good antioxidant agent due to the presence of 2-hydroxyl group. Thirdly, the secondary metabolite present in the plant known as Micromelum, and the class of the metabolite is shikimic acid. The fourth and the final secondary metabolite extracted from the plant known as marmazine glycoside, and the class of the metabolite is fatty acid. Additional note, the root word marmazine is a chemical compound precursor in cerulean and linear furanocoumarine biosynthesis.
So today I will continue on the presentation on biosynthesis pathway of the selected compounds. So this is the pathway on how the sesame bio pathway look like. Okay, first, the pathway branches out from a common cascade of phenylpropanoids by stereoselective radical coupling of 2 molecules of a monolignal coniferyl alcohol guided by a dirigent protein. This coupling reaction results in the formation of the enantiomer of pinoresinol, a central precursor of lignans. Then, pinoresinol is then converted to sesamine via sequential oxygenation, resulting in the formation of 2-methylene dioxide bridge. This reaction is catalyzed by a single cytochrome P450 monooxygenase CYP81Q1 that is also known as piperito or sesamine synthase. The last one is, in contrast, the terminal metabolism of sesame lignans, particularly sesamolin and sesaminol, remain enigmatic, with neither the type of oxygenation nor the putative substrate being well understood. The second one is marmosine glycoside. So we can see here, this is the figure on how the biosynthesis pathway of marmosine glycoside look like. Okay, the first one is umbelliferone is considered the precursor of furanocoumarins. It is first prenated in the 6 for linear furanocoumarins or 8 position for angular furanocoumarins to yield the methyl subrosine or ostenol respectively. Next is the dimethyl allyl diphosphate is required for the 6 prenylation. And the last one, dimethyl subrosine is transformed into marmosine by marmosine synthase, that is cytochrome P450, which catalyzes the instant cyclization without releasing an intermediate. Presumably, it is due to the deliocalization of the double bond electrons by the 7-hydroxy group, forming the dihydrofuran ring from orthoprenylated phenol. And the last one is micromelin. This is how the pathway of micromelin looks like. Okay, flavonoids are hydroselected phenolic substances and are known to be synthesized by plants. The functional hydroxyl groups in flavonoids mediate their antioxidant effects by scavenging free radicals and/or by chelating metal ions. The chelation of metals could be crucial in the prevention of radical generation which damage target biomolecules. Coumarins are a group of compounds derived from the phenylpropanoid pathway. They possess a 2-benzopyranone core structure, and individual members of this class of compounds exhibit different substitution patterns. That is the difference between hydromicromelinin and micromelinin. So that's all from my parts. I will pass it to the next presenter. Thank you.
Next, the medicinal usage of secondary metabolite. Firstly, it's involved in the production of streptomycin. It's used in the treatment of diseases such as tuberculosis, tularemia, and in the veterinary medicine for use against gram-negative bacteria in large animals. Secondly, it acts as an antifungal, antimicrobial, antiprotozoal agent. It's used to treat infection caused by microorganisms, including fungi and protozoa. Thirdly, it's used in the production of vitamins, non-riboflavin, L-sobose, and vitamin B12. The fourth and the final medicinal usage of this plant is involved in the production of ergotamine. It's used to treat patients with migraine and can increase uterine contractility therapeutically immediately postpartum to decrease uterine bleeding.